Hi, good afternoon, everyone, and welcome to our webinar Wednesday. Today's webinar, HPA Access Stress, Controversies, Conundrums, and Opportunities. My name is Amy Paoletti, and I'm a member of the Dutch Education and Marketing team, and I'd like to thank everyone for taking time this afternoon. I know a lot of you join us every month for our webinar Wednesdays. We really appreciate that, and we hope you get a lot out of it. Today's webinar, I just wanted to discuss a few bullet points of what we hope you get from this. Discuss how adrenal fatigue incorrectly frames HPA axis dysfunction by reviewing how stressors control brain function. We hope to discuss expanding your understanding of the role of HPA axis in metabolic and circadian signaling. We are going to discuss a little bit about debunking the notion of the pregnenolone steal. I know a lot of you guys learn that in other courses that you take. As an explanation for low DHEA or other hormones in stressed individuals. And then also provide an overview, a valuable resource of understanding the HPA axis and therapeutic options for stress management. So just a few housekeeping items before I introduce Dr. Williams. We're going to answer as many questions as we can at the end of the webinar, so pop your questions into the chat. If you miss anything today, if for some reason you have to pop off or you've got technical difficulties that we can't resolve, we will send the on-demand recording to all of you tomorrow along with the presentation slides. So I'd like to welcome Dr. Thomas Williams with us today. Really excited to have him join us and hope that we can have him join us again later on this year. Dr. Williams earned his doctorate from the Medical College of Wisconsin, where he studied molecular immunology in the microbiology department. Since 1996, he has spent his time studying mechanisms and actions of natural-based therapies and is an expert in the therapeutic uses of nutritional supplements. He was a longtime VP of Science and Regulatory Affairs for orthomolecular products, where he developed a wide array of products and programs which allow clinicians to use nutritional supplements and lifestyle interventions as a safe, evidence-based way and effective tools for a variety of patients. Tom teaches at the University of Wisconsin School of Pharmacy, where he holds an appointment as an assistant adjunct professor, as well at the University of Minnesota. So go ahead and take it away, Dr. Williams.
Thanks, Amy, and thanks for the whole Dutch team for inviting me, and welcome everyone. As I start, I'm thinking, I think of stress, and obviously the last year has been very stressful for many years, and we have this huge thing going on in the U.S. with. I just talked to somebody down in Texas, and so many people are under stress right now. So I'm mindful of all those people that are under a tremendous amount of stress as we're thinking about the stress response system. So this is a topic that I've been teaching on and thinking about and obviously writing about for over a decade now. And I like to call this sort of like my stressful mission. So I'll kind of give you what my mission has been thinking through this as we begin this. So one of the things that I've been doing is trying to realign the functional medicine community to the current research of HPA axis and stress dysfunction. And when I got into this, as you'll see, the whole idea of the stress response system was kind of in a different place, let's say, 15 years ago. And as we've been sort of climbing out of those changes, we're trying to realign that to expand the view of the HPA axis beyond the stress response. That's something that's really more new to me. I'm teaching it more recently, is after we've corrected the idea of the HPA axis and stress dysfunction, really to understand the HPA axis has a lot more to do with what happens in the body than just quote unquote the stress response itself. Part of that has been to try to create a common nomenclature around stress and HPA axis function that's both useful and accurate. So we're going to talk a little bit about that. Starting back in 2007, trying to encourage more uniformity in how lab testing is done for the HPA axis, both in the units, the nomenclature, the collection, the instructions. It was really a chaotic mess, if I could use that term, a decade ago. And I think we're starting to see a lot more, and I commend Mark and the Dutch team for for really, looking into this in ways that, a lot of other people have not, and it really shows in what they've done. To promote more research related to testing and HPA axis intervention. Surprisingly, there's little in the way of therapeutic treatments related to a lot of the traditional ways that functional medicine clinicians measure stress and the HPA axis. And then finally, to help clinicians incorporate therapies that are directed specifically towards the real root of the stress or the HPA axis dysfunction that they're seeing. So one of the things I've been spending a lot of time debunking or trying to get people to think differently about is the fact that when we think of stress, somehow we always think of adrenals, and we even say to people, you have adrenal issues, as if that is their problem. And so at some point, you know, not in the not too distant past, this whole thing became about adrenals. Well, obviously, there was a book that had a lot to do with this, and I think Jim Wilson did a great job of helping us understand that there's more to chronic disease than just or trying to connect the sense of the stress response to chronic disease. But unfortunately, the terminology adrenal fatigue has been taken and become sort of the definition of what the stress response or a dysfunction of the stress response. And he's not the only one. This has spawned a whole lot of people to view everything as being adrenal fatigue or adrenal related. And so what I'm hoping to do, hopefully some of you already climbed out of that. If you've been listening to the webinars that Dutch has been putting on, then you clearly probably have already migrated away from that. But I think it's important to to continue that approach and to think why that's so important. So when we start talking about adrenals, so first of all, the whole idea is oversimplified and primarily mostly wrong. And that's probably the biggest problem. And because of that, it limits the leverage of real solutions. If the solution is not, or if the problem is not the adrenal gland, then the solution is not, i.e. supporting the adrenal gland. And so it's really important that we begin to climb out of this idea that stress-related phenomena is adrenal related or functions based on the adrenal gland. So why is it oversimplified? Well, adrenal function or dysfunction is rarely a defining feature or root cause of the stress response system's dysfunction. That is, if you think of like the pancreas that begins to wear out as it needs to produce more and more insulin in a type 2 diabetic with low insulin sensitivity, you actually do get a wearing out of that gland in the production of insulin. But that's not the case here. We don't wear out the adrenal gland and its ability to produce cortisol, and that's why the cortisol is reduced. So this oversimplification misses really the root cause of these phenomena. And it's under leveraged because, like I said, the most powerful therapies that clinicians can use for supporting the stress response or the HPA axis are not really adrenal support. Although some may cross over to helping the adrenal gland function, most of them are not. So the real solutions are often overlooked or under leveraged because of that. So the other thing is we have sort of a campaign against the term. Maybe some of you know that the Hormone Foundation Endocrine Society has specifically published, on their website, these pages that they send out trying to warn people against the term adrenal fatigue. They say it's not a real medical condition. There's no scientific facts to support the theory that long-term mental and emotional physical stress drains the adrenal glands and causes many common symptoms. There's no test that can detect adrenal fatigue. Supplements and vitamins made to, quote, treat adrenal fatigue may not be safe. Taking these supplements when you don't need them can cause your adrenal glands to stop working and may put your life in danger. Obviously, a little hyperbole there. But nonetheless, they're starting to focus on this terminology, and they say at the bottom here, you can see doctors urge you not to waste precious time accepting an unproven diagnosis such as adrenal fatigue if you feel tired, weak, or depressed. So obviously they've gone overboard in kind of attacking this idea, But in the published literature, we do have people writing papers in the peer-reviewed literature, like this one, adrenal fatigue does not exist, a systematic review. Actually, if you look at that, it's actually not a systematic review of adrenal fatigue. It's a systematic review of what they thought adrenal fatigue was. So it's actually very poorly written, but nonetheless, they're using that terminology. I like the one from the South African Medical Journal, we are tired of adrenal fatigue. So pretty much the only place you find adrenal fatigue in the literature, in the peer-reviewed literature, is oftentimes in kind of critiquing the concept as being non-existent. So are these critiques right? Is the Endocrine Society right? Well, I'd say partly. The term adrenal fatigue does not properly describe the stress response that leads to change in adrenal hormone output or a sense of fatigue. So I think we should, probably begin moving away from this terminology. We should really stop using this idea because it really is not helpful. So in that respect, I think the Endocrine Society is right. However, they're very incorrect in their assertions that, in the first point, that long-term stress has no effect on adrenal hormone output, or that no test is capable of assessing the effects of stress on human physiology. And they're certainly wrong about the fact that supplements will, all of a sudden cause your adrenal glands to stop and you could die. And obviously that's there's no evidence for that. So in order to correct this, we need to rethink about how the adrenals, or how we use the concept of adrenals within the HPA axis. As you'll see, obviously, if it's the HPA axis, the A is still adrenal. So we're not trying to completely remove it from the equation. And obviously, if we're measuring cortisol, it's clearly part of the whole process, but we need to really be thinking about the HPA axis more comprehensively. So I'm kind of a guy that believes nomenclature matters, because how you define things is how you approach them. Whether you like it or not, when you use terms, they really in many ways affect the way you look at things. So when we think of primary adrenal insufficiency, this is a real, obviously it's not very common, but it is a real, but it's not stress related. It's probably autoimmune related, but it's not a stress related phenomena that causes primary adrenal insufficiency. So unfortunately, in the nomenclature of adrenal diseases, we will see that really only primary adrenal insufficiency is actually an adrenal disease or adrenal caused phenomena. Secondary and tertiary adrenal insufficiency actually is a brain derived phenomenon, not an adrenal derived phenomenon. So stress related changes in adrenal hormone output are regulated by the HPA axis, usually by adaptations, primarily and almost exclusively in the brain. So when we see changes in cortisol, or in many cases DHEA, these are caused by changes in the brain. They're not caused by the fatigue or the inability to produce hormones by the adrenal gland. The adrenal gland basically gets its signal from ACTH, which comes from the pituitary. And like I said, secondary and tertiary adrenal insufficiency are also hypothalamic or pituitary issues, not an issue of the adrenal gland. So that's an unfortunate nomenclature issue within traditional medicine that also gives some confusion because those conditions are not driven by the adrenal gland either. So again, I think most everybody here probably knows the adrenal glands respond to the brain. And so we have a variety of stress signals from outside the hypothalamus, along with feedback signals of cortisol at the hypothalamus and the pituitary, which then determines how much ACTH signal and subsequent adrenal cortisol output. So the adrenal gland is responding. It's a responsive organ. It's not controlling the situation. The only way that it controls it is by the feedback inhibition of the amount of cortisol it produces, but that's a very passive way of thinking about it rather than active. So as we said, in a normal subject, you have the signal that comes CRH or CRF from the hypothalamus and ACTH from the pituitary, and then you get this feedback inhibition both at the pituitary level and the hypothalamus. So the signal, we always think of the HPA going down, and we know that there's this feedback inhibition, but as it turns out, you can, by changing the feedback inhibition, in the case with patients with major depression, this feedback inhibition seems to be down-regulated. And we won't have time to talk about perhaps why this is, but it's likely due to neurotransmitters neurosteroids, and maybe even the endocannabinoid system, which alters the receptor sensitivities in the brain. But nonetheless, because that's toned down, the brain then says, okay, I need a bigger signal. And so you see oftentimes people with major depression with elevated cortisol because they're not getting that feedback inhibition is not strong. In the case with patients with PTSD, this is typically adults with PTSD, they will have an oversensitive or a hypersensitive feedback sensitivity, and that will then downregulate ACTH, which will then cause them to have low cortisol levels. Oftentimes, they will also have elevated DHEA, and it appears that their body's trying to protect themselves from too much cortisol. So what is the correct terminology? Well, I used to just say this was HPA axis dysfunction. I'm now using the term stress-induced HPA axis dysfunction because that's really what we're talking about, is changes to the HPA axis that are induced by stress. Obviously, it can be driven by other phenomena, perhaps, but this is what we're talking about: stress-induced HPA. But sometimes people call it maladaptation to stress, or a stress response dysfunction, or simply an adaptation to stress with consequences. So as it turns out, the brain is continually adapting to its surroundings, and if there are major stressors in the environment, the body begins to adapt to them. And sometimes it does that well, sometimes it does it with consequences over time. So, many years ago, George Crusos put together this review. It's still probably the best one that is this comprehensive, where you can find all kinds of disorders that are associated with either an increased activity of the HPA axis or a decreased activity of the HPA axis. Sometimes people move from one, or at one point in time where you have an increased activity, and then eventually as the system begins down regulating to a condition on the right hand side, which is decreased activity. So we know that the HPA axis over time is being controlled or it being associated with many conditions. We don't always have a cause and effect. Which one is first? Is the stress response changes first, or is the chronic disease condition first? But nonetheless, we know that there's a relationship with, I would say there's probably a relationship with every chronic disease condition, but we don't necessarily know of or can't measure that in every one. So these are the most appropriate terms. And then obviously within that, there are very specific terms. So if somebody has low cortisol or high cortisol, we can just say it's hypocortisol or hypercortisolism. Sometimes, we'll see later, or maybe we won't have time to talk about it now, but the idea of the staging, stage 1 or stage 2 or these kind of things, we don't always know what's happening, but we can know by measuring whether they're high or low cortisol. Obviously, these terms are also used by endocrinologists to define Cushing's and Addison's. So it's a little bit tricky sometimes to use those terms because other people use them differently. But if you have low DHEA or DHEA sulfate, and in the literature, you'll see things like high allostasis or high allostatic load or burnout, and burnout can be, there's certain definitions used in the literature that are more appropriate for HPA axis burnout than other types of let's say, burnout. But if you ask the question, what is going on? What is the HPA axis trying to do? What is as it were, the goal of the stress response? Essentially, it is to maintain effective blood supply of oxygen and nutrition to the brain, to the heart, to the skeletal muscles for an immediate survival. So this is what the stress response is. We typically think of it. You have sort of the autonomic, quick acting adrenal production of norepinephrine and epinephrine, and then later you have the effects of the HPA axis, which is produce increased energy production by recruiting substrates like glucose, fatty acids, amino acids. From body stores, you're going to enhance gluconeogenesis. You're trying to optimize your ATP production for short-term needs. And we're thinking of, immediate needs within the next minute, but also the HPA axis, we're thinking of the next, let's say, 2 to 4 hours. So can I survive this event without dying or without, coming into major injury? And it often does this at expense of long-term metabolic functions. So the idea that I often use, or the terminology that I sometimes use, is achieving physiological resilience at the expense of metabolic reserve. It's going to pull the reserve capacities from various tissues to survive now. if you know Bruce Ames's triage theory about how cells do this with nutrients, it's the same sort of a general idea. And like I tell people is that stress drives on the same road that all your metabolism needs. So just like an emergency vehicle, this one actually is in Germany. You can see by the word there, but you know, everyone needs to get off the road so that the emergency vehicles can come through. Well, that's in some sense, if you're constantly trying to respond to stressors, you're trying to push aside normal metabolic functions or trying to use normal metabolic functions alongside the stress response. And the reason that is because the HPA axis is the body's normal metabolic regulator. So as it turns out, you kind of want to use the normal regulators, but now you have sort of a heightened uh, urgency of those same functions. And as it turns out, that's where some of the consequences come in. So for those of you who don't know, I just recently published the second edition of this book, the role of stress in the HPA axis and chronic disease management. The first one was published in 2015, and we just expanded it. Not only did we expand each of the section, updated the references, but we added a section on the endocannabinoid system, the memory and brain function, and also how the stress affects the thyroid function, and then we expanded quite a bit of the therapeutic section as well. And so if you're, a lot of what I'm going to talk about obviously is from the book here, and a lot of the images that I'm going to use come from the book. So you can get that, and we can send it to you, or there's actually an ebook version as well. So I think most of the people, hopefully most of the people on this webinar have some idea of the stress response. We're going to spend most of our time talking about the HPA axis on the right-hand side, but you can see epinephrine and norepinephrine being signaled. This happens obviously very quickly within seconds. I don't have time to talk about it, but the brain has its own ability to produce norepinephrine. It's one of the reasons why, you know, you can't wait for the epinephrine, norepinephrine to be produced by the adrenal glands and get all the way to the brain. So the brain has its own ability to produce norepinephrine almost instantaneously. But nonetheless, the other signals, the signals for CRH or CRF from the hypothalamus and ACTH are a little bit slower, take a few minutes to respond, and then we get the production of glucocorticoids. We think of cortisol in humans. There's also, obviously, the adrenal gland also produces DHEA, some of which is in relation to a stress response, as well as mineralocorticoids, aldosterone, which and there's a lot of ability for cortisol and aldosterone to bind to each other's receptors. So there's a lot of things going on when we think of the HPA axis, just when we back up and look at it like this. So one of the things that I'm going to focus on primarily today, and it's going to be a little bit later, is like what makes the brain stressed. So the hypothalamus is actually it's not one nerve bundle, it's a series of nerve bundles or a groupings of nerve bundle. We call them nuclei. And they are there to basically take account. I call it like a surveillance system, surveilling the whole body for all kinds of different, what is your temperature, what is your hydration level, what is the inflammatory signals in your body, what is the blood glucose level, it's a major one, and then takes all that internal information and then also takes all the external information, everything your body is sensing outside of you, everything you can see, everything you can feel, even sort of your memory or your premonitions or your worries, all of that goes in and gets consolidated into the paraventricular nucleus, which I show here right in the middle of the screen. That's the consolidator of all this information. And then I also show the suprachiasmatic nucleus, which is this little tiny nuclei of nerve bundles right above the optic chiasm. And essentially that allows light and the light that comes in through the eye to be able to help create a circadian component to this signal. So those are the two that we think of as being the most profound in how much and the timing of the CRH that comes from the hypothalamus and travels down to the pituitary gland. So you can see on the left-hand side, maybe this is a little hard to see, but you can see that I have here the suprachiasmatic nucleus, the SCN, and then the higher orders of the brain, the CNS. They feed into the paraventricular nucleus, which then triggers, the release of ACTH through on the anterior pituitary. And that basically happens, as you can see on the right-hand side, by the release of CRH. CRH binds to a CRH receptor, and I'm showing here now, this is obviously a cartoon image on the right-hand side of a corticotroph cell in the anterior pituitary, but you can see that CRH binds to the CRH receptor. Of course, those of you who know that arginine vasopressin is also another sort of CRF, or another releasing hormone that comes from the hypothalamus. It also has an effect, and I'm not going to go into that because the primary signal that triggers ACTH release is CRH or CRF. We use them both somewhat interchangeably, and then we get this signal that happens. But as it turns out, there's a lot of research over the last decade or more trying to understand. We always think of CRH or CRF as having just binding to its receptor and doing its thing. As it turns out, there's a whole family of urocortins for which CRH is one, and there's actually several, in this case really two, but maybe even a third receptor for CRH. So as it turns out, there are multiple urocortins, and you can see here they bind differentially to these. And of course, CRH1 binds CRF very well, or CRH very well, and that's where we get ACTH. But as we look into other tissues, you'll notice that there are all kinds of receptors that allow for the binding of both urocortins and CRH, and they're not all found in the pituitary. You can see here, if we look at the centrally distributed ones, you'll see these are all over the brain. The anterior pituitary is listed here, but the locus coeruleus, which is responsible for the release of norepinephrine in the brain, and you can see higher orders of the brain like the hippocampus and the amygdala, and also CRH2 receptors, which are binding urocortins. But look down below, we see CRH and receptors on the adrenal gland, in the skin, in the GI tract, in the peripheral vasculature. So as it turns out, we're learning that this common molecule that we think of just as being regulating the stress response is actually responding, is affecting the immune system, is affecting the skin, is affecting the GI tract, and perhaps a number of other things that we don't know. So this is kind of interesting, and like I said, I don't want to get off on a tangent, but as it turns out, CRH is not just, CRH receptors are not only found in the immune cells, but CRH production, that is, mast cells and macrophages can actually produce CRH. So you might have actually local production of CRH within immune cells that are actually helping to regulate the immune system. And it tells us that there are parallel systems of surveillance. Now, I often talk about the fact that the HPA axis, the immune system, and the GI system are the surveillance systems of the body. They're highly coordinated, and one of the things they all have in common is this ability to produce and be sensitive to CRH. So just because you have a CRH receptor doesn't mean that CRH may come from the brain. It may come from other tissues. Interestingly, and this is something I'm really interested in digging into at some point, is that the skin has its own HPA axis. And by that, I mean, within the skin, we have the ability to produce CRH, ACTH, and because we have steroidogenic cells within the skin, also cortisol. And so it's almost like the skin has its own, I would say, stress response system. Obviously, it doesn't have a hypothalamus and pituitary or adrenal, so it's not an HPA, but it's almost like an HPA-like stress response system that's self-contained within the skin itself. Again, this is very interesting. Obviously, the immune system is very active in the skin as well, but it's something that we need to begin thinking about as we start looking beyond just the HPA axis when it comes to what's going on, surveillance, metabolism, and modulating these kinds of things. So let's just talk about the adrenal. If it's all about the adrenals, what are we talking about? Well, there's not just one gland in the adrenal gland. There's multiple glands. The adrenal medulla, as you can see at the bottom, that's where we produce epinephrine and norepinephrine. That's almost a different glandular system altogether than the cortex, which itself is made up of three layers of tissue, all of which are derived differently from embryogenesis, the zona glomerulosa, the zona fasciculata, and the zona reticularis, obviously producing respectively aldosterone, cortisol, and DHEA or DHEA sulfate. So when we start thinking about this, it's really important to realize that we're really talking about a gland that is regulated in a multi-dimensional fashion. And the reason that's important is because when I teach adrenal steroidogenesis, now remember, this is adrenal steroidogenesis. So in order to do that, we need to divide these into three different groups. So the zona glomerulosa, fasciculata, reticularis all have the ability, all are steroidogenic, and a steroidogenic cell is essentially a cell that is capable of converting cholesterol into pregnenolone. So you have to be able to take cholesterol and produce pregnenolone. And from that, of course, that happens in the mitochondria. And once the pregnenolone is made, then depending on the enzymatic machinery in that cell, it's able to produce either aldosterone or cortisol or corticosterone or DHEA or some of its derivatives, the androgens. And so this is the reason this is important is because oftentimes when you have studied or when somebody has taught you steroidogenesis, you see the traditional way of thinking. It starts with cholesterol, pregnenolone, and then it goes through all of these different pathways. And it's true, that's one good way to teach it, but unfortunately, we often teach it, DHEA is on there, testosterone is on there, estrogens are on there, and it's this big, big metabolic pathway. The problem is these cells don't share precursors. So the notion of pregnenolone steal, so the idea that we've often had is that if the precursor to all of these hormones is pregnenolone, and we have this big huge pool of pregnenolone, and that's the first um, first hormone that's produced, then if I need to produce lots of cortisol, I'm going to steal away all that pregnenolone, and the poor body is not going to be able to produce estrogen. It's not going to be able to produce testosterone or DHEA. And this is often referred to as pregnenolone steal. And so I often say this is a precautionary tale. When we often use simplistic diagrams of steroidogenesis, we get this notion that that actually could occur. But I can give you a number of different reasons, but there's no way that the mitochondrial produced pregnenolone in the zona reticularis gets shuttled over to the fasciculata to produce cortisol or vice versa. So that really doesn't happen. Physiologically, that doesn't happen. So we don't have that going on. There's a number of other reasons, mathematical reasons. By the way, DHEA circulates 500 to 1,000-fold higher concentration than cortisol. So even the idea of pulling away some of that pregnenolone, even if it were possible, really mathematically wouldn't have much of an effect. So here again is another terminology or another explanation that we give that sounds good if we show sort of a normal or I should say a simplistic steroidogenic diagram, but when you see it like this, you realize that that's really not the case. Now, we don't have time to get into the details, but it is often true that when under stress, where cortisol goes up and DHEA does go down. And there are many phenomena, there are many reasons why, perhaps why that is, but not because of pregnenolone steal. So the other thing I spend quite a bit of time on, and this is something that we're not going to get into detail here. I spend in my book, I spend a significant amount of time on this, is that we often end our conversation when we say, okay, how much cortisol is this person producing? And we just imagine if we just measure that, we know everything. Well, as it turns out, there's a lot of things that affect how a particular cell or tissue responds to cortisol. It's highly dependent on a number of factors, many of which are independent of the actual amount of cortisol released by the adrenal gland. So some of those are systemic, so maybe affecting cortisol everywhere, like cortisol availability, cortisol binding globulin, and then many of these are tissue specific. So again, I'll just briefly show what these 7 are. So the rate of cortisol metabolism, the bound versus free cortisol. So as most of you know, corticosteroid binding globulin binds almost 95% or more. So the body has sort of a a reservoir of available cortisol, but most of it's bound, um, and that actually can be released by inflammatory processes on a local level, meaning if one tissue needs more cortisol, it can actually release some of that from the binding globulin, and you can have some anti-inflammatory activity on a local level. Um, the enzyme 11 beta hydroxysteroid dehydrogenase, there's two of them that interconvert cortisol to cortisone, the active versus the inactive form. There's the genomic signaling within the cell, and we're not going to get into, again, the details of this, but there are a lot of polymorphisms that change the sensitivity of glucose, of the glucocorticoid receptor to cortisol, some of which is managed by insulin sensitivity and glucose. Heat shock proteins, which are really important for shuttling the glucocorticoid receptor in and out of the nucleus. And we've learned a lot in the last 10 years about different heat shock proteins and what we call co-chaperones that, depending on how much you produce of heat shock proteins or these co-chaperones, has a major effect on your stress resiliency. There's non-genomic signals, and of course, DHEA. And this is just a graphic illustration of those. Again, like I say, in the book, I actually go through each of these in a lot of detail. But they have a lot to do with how a person may respond to the cortisol levels that they're producing. In the case of the brain, obviously the brain is also a target tissue. So many of these signals are effective in the brain, and they're affecting the feedback inhibition. So as these are modulated in the brain, it's going to affect the feedback inhibition, which will eventually change the cortisol level produced by the adrenal gland. So as you can see, there's a multi-level adaptation at peripheral tissues, at the brain, that affect not only the amount of cortisol, but really more importantly for your patient, what is that cortisol actually doing to the tissues? What is going on? What is it driving? And so as I've been getting into this, I've been asking the question, well, how about the cortisol in the mitochondria? When I first started researching this years ago, there was a question about whether the mitochondria had glucocorticoid receptors. And the answer was we didn't know. And it looks from the DNA. If you just look at the DNA and the expression in the mitochondria, it doesn't appear they have traditional glucocorticoid receptor. But as it turns out, they do respond to cortisol. So the reason that's important is because mitochondria are highly important for the metabolism in the cells. So there's a number of these are papers that reviewed this. Bruce McEwen, who's now passed away, was leading this effort. And he had this great title, mitochondrial allostatic load, putting the gluc back in glucocorticoid. Remember, what is a glucocorticoid? It is a glucose regulating hormone. And so we sometimes forget the fact that glucose regulation, or I would say energy regulation, is really, really important. And as it turns out, chronic stress and acute stress have effects on mitochondrial function. It shouldn't really surprise us, really. It not only changes their effectiveness, but it actually changes their structure in many ways. So normal mitochondria, they go through this fusion, fission, depending on how much energy and what their own stress levels are. Typically, these larger mitochondria, we think of them as, let's say, more relaxed, perhaps. They go under stress, they become metabolically stressed as well. They go into this fission where they produce smaller mitochondria, and which ends up causing more reactive oxygen species damage, less ATP production, and creates sort of the cellular dysfunction that we think of when we think of the metabolic stress on cells. So we have the reason we can connect metabolic stress, again, is because, remember, I told you the HPA axis we think of as stress is driving in metabolic function. And so it basically creates an increased metabolic stressors on the cells. And we think of that typically as oxidative stress. So if you just look physically and you think of this, acute stress, many times the HPA axis and the cells are really good at dealing with acute stress. They have systems in place that are designed to deal with things quickly. So we can increase the mitochondrial biogenesis increase replication, increase ATP production, and quench free radicals. But if that stress becomes chronic and it's not able to replete or build up its reserves, and you start reducing the reserve capacity, all of the opposite happens. You reduce mitochondrial mitogenesis, reduce ATP production, increase reactive oxygen species production. So in some ways, if you think of stress, one way of thinking of the generic term of stress, whether it's metabolic or tissue stress or HPA stress, it's a depletion of the metabolic reserve to prevent us from being resilient for the next metabolic challenge. Okay, so if you deplete the metabolic reserve, you can't respond the next time you have a challenge, and then you start getting consequences. So that is the consequences of stress are due to the depletion of metabolic reserves. So what is the solution to treating patients with HPA axis dysfunction? So if it's not about the adrenals, then we probably should stop thinking about how we can fix the adrenals and ask a simple question: what makes the brain stressed? So I've done some teaching on this for a while, and this figure is one that I have taken. I've slightly modified it, but it's essentially very similar. You can see here circadian disruption, perceived stress, glycemic dysregulation, and inflammatory signaling. Now, almost everything that the brain would interpret as a stressor would come from one of these four categories. Now, obviously, there are subtle other things, but if we're going to consider all the things that are modifiable and interpretable, these really categorize almost everything that you can do to understand what the brain sees as a stressor. So if you're gonna have a program that allows you to deal with these patients, you really need to have a successful strategy for assessing these. So you need to be able to assess somebody's perceived stress or their circadian rhythm, their inflammatory signal, and their glycemic dysregulation. You need to have lifestyle and nutrient therapies to reduce these key areas that are driving the HPA axis. And then you need to connect where you can with adrenal hormone output. So obviously, one of the reasons why having these tests and the different ways that you can test cortisol and DHEA, different timings, and and even what Mark and his team are doing with different urinary metabolites, and get the more information that you can get that connects what's going on with the HPA axis with some of these outcomes, the better you are at being able to target what strategies are going to work in different patients. So let me just go through a few of these. So what makes the brain stressed? When we think of perceived stress, we think of mental and emotional. So, the mental and emotional stress is a big part of this, but also it's the interpretation of the mental and emotional stress within the brain. And again, we don't have time to talk about this in detail today, but neurotransmitters, neurosteroids, and the endocannabinoid system are part of the interpretive strategy or the interpretive milieu of the brain in dealing with a mental and emotional stressor. So, and also the feedback inhibition of cortisol itself. So we don't have time to talk about that, but in this, you can see in this picture here that these are related to one another in how perceived stress. It's really a mashup of mental and emotional. The pictures probably should show the mental and emotional arrow going through the anxiety, the neurotransmitters and neurosteroids, because that's really what produces the perceived stress. So what is perceived stress? Essentially, it's converting external events into internal signals. What you perceive and what I perceive as stressors are different. So public speaking, as we know, is one of the most stressful events, but it's not the act of public speaking, it's the perception of the event itself. And so all of these things are really related. It has a lot to do with your history, a lot to do with your memories of events. And stress researchers have determined that these are the four categories, the four things that make up a perceived stressor. Something that's new, something that's unpredictable, something that you sense is a threat to yourself, either physically or your ego or how people will perceive you, and a lack of control. In fact, in some ways, control becomes sort of the key word. Anything that you don't believe that you are in control of you will have some stress related. Your perceived stress will go up. And so it's one of the reasons why the perceived stress questionnaires almost all have to do with questions like, do you feel like you can control this part of your life, your finances, your relationship, your job, these kind of things. And so that's really important. For those of you who are not aware of the term neurosteroids, let me just introduce it briefly and say that the brain, remember we talked about steroidogenic cells. Well, as it turns out, the brain is a steroidogenic tissue. So the several brain tissues can produce their own DHEA, their own pregnenolone, their own, there's actually another set that you can see, allopregnanolone. There's actually several different hormones that would fall under the neurosteroids. So the brain can produce some of these, including the free and the sulfated form of several of these, and they can affect GABA receptors and affect signaling within the brain that affects plasticity, that affects muscle relaxation, and a number of other things, but also affects the interpretation of the stress signal itself. So this is what I'm talking about. I don't, again, the endocannabinoid system can do this as well. The endocannabinoid system can change the sensitivity of the brain tissue to stressors. So this is what I mean when I'm talking about neurotransmitter and neurosteroids. They're all together affecting this. So circadian disruption. Circadian disruption is something I've kind of focused on quite a bit over the last couple years. And of course, we think of, circadian disruptors. If you have sleep deprivation, even for a single night, this actually can be shown to affect the HPA axis, but certainly chronic short sleep or disrupted sleep is going to be a problem. We know jet lag, daylight savings, anything that would be phase shifting or shift work, which is like a major phase shifter. If a person is working third shift or swing shift and they are not precise and diligent in maintaining a daylight cycle, even if it's the wrong daylight cycle, if they're not diligent in maintaining that, they are going to constantly be in battle with their HPA axis. Because remember, as we're going to get back to here, I'll show you in a minute, the HPA axis is highly related or highly synchronous with the light dark cycle. So depending on what you know about this, I'm going to briefly talk about circadian rhythm. And you can see here that circadian rhythm is driven primarily by two different clocks. A clock that is found in each of the almost every cell in the body. We call it the peripheral clock. This is a clock that runs, a metabolic clock that runs within tissues. And then the brain, or the central clock, which comes from the suprachiasmatic nucleus, which is synchronized or supposed to be synchronized with sunlight coming through the optic nerve. And essentially, the molecular clock, we will call it, I'll show you here in a minute, is just simply a transcription loop, transcription translation loop in the tissues. So for those, again, who are not as familiar with this, I'm just going to talk about this briefly. Within each cell, the peripheral clocks basically run like this. You have a series of proteins. You can see here they're all given different names: per-cry, ROR, RevRB. I'm not going to talk about each one of these. BMAL and clock. So essentially what happens is the cell produce these proteins, and these proteins are transcription factors. So they produce these proteins and they come back in and begin regulating other proteins. And when they regulate those proteins, you can see there's certain cycles. They affect each other. So as it turns out, the way that the half-life of these proteins is about 24 hours. So there's a 24-hour cycle that constantly runs because these proteins are produced and then they come back in the body, either inhibit or encourage other proteins to be produced, and you get this transcription cycle loop. And along with that, you have other cells, or excuse me, other proteins, other genes that are transcribed, and those we call the clock genes, or the clock control genes at the bottom. And you can see those produce all kinds of proteins. So this cycle is running in the background, and it's causing a number of different genes. Maybe some people say 30 to 50% of mammalian genes are in some way controlled by the circadian rhythm. And this cycle needs to run very, very specifically in these cells. But obviously, the cycle is not exactly 24 hours and can be influenced by other things. So there needs to be other signals that come from the brain to tell it how to keep on track. And those other signals that come from the brain, obviously light is the biggest one. You can see that's got the largest arrow, but also feeding cues, eating, and activity. All of these things are intended to resynchronize all of the physiology of the cells. And probably the other one that we don't think of, we don't exactly know exactly how, but the gut microbiota is likely also giving signals either to the brain or to the periphery or both. So that's probably the other signal that is not often thought about in that process. So as it turns out, most of these signals come from the suprachiasmatic nucleus, not surprisingly. So the brain controls feeding and hunger. The brain controls glucocorticoids. The brain controls body temperature, which is also a circadian function, and also autonomic innervation. So direct innervation between the suprachiasmatic nucleus and certain tissues, especially the adrenal gland. So as we'll see here, the suprachiasmatic nucleus has a direct innervation with the adrenal gland. So we're going to talk about these two things. So let's go back to this picture that I showed earlier, and you can see the suprachiasmatic nucleus acts as the main, what's called Zeitgeber, which is the German word for timekeeper. And it signals through, obviously, like we had, the ACTH and cortisol. So we have glucocorticoid being produced, but we also have the circadian rhythm being, you can see this dashed line between the suprachiasmatic nucleus and the adrenal cortex. So there's a way that that functions, and we'll see here in a minute, that affects the cortisol awakening response to help the adrenal gland know, because the adrenal gland doesn't have its own eyes, but the adrenal gland needs to know exactly when a person wakes up. So if you were to measure ACTH and cortisol, you certainly wouldn't want to see something like this. But this is actually what would happen if you were measuring cortisol literally every minute or every few minutes of the day. It's kind of a jagged curve like this. Most of you see something more like this, where it's smooth, whether or not you're taking the cortisol awakening response, which is that spike in the middle, or just the 4 points on the right-hand side. Hopefully, most of you are using the car. But nonetheless, it's a smoother curve, or we'd like to see it as a smoother curve. But there's a lot of things going on, and that's because there's all kinds of adjustments going on. We call it an ultradian rhythm. If you really know what's going on, there's all of these little rhythms that are going on all the time. And that's because there's a back and forth relationship between the clock systems and the HPA axis. And so there's this feedback that's going back and forth between changing sensitivities to ACTH in the adrenal gland, which are lowest in the morning and highest later on. And then we have cortisol affecting the clock genes that I just showed you. So the relationship goes back and forth. Obviously, the suprachiasmatic nucleus is also is affecting both sides of the program. So again, it's a little hard to do this on a webinar, but this is basically showing a little more of the details of how the glucocorticoid receptors, obviously, which are binding cortisol, have an effect directly on the clock genes. Remember I said the clock genes are just transcription cycles or loops. And as it turns out, not only do cry and purr and BMAL and clock, some of the proteins that I talked about bind to and turn on or turn off these genes, but glucocorticoid receptors can also do the same thing. So glucocorticoid receptors modulate the peripheral clocks by increasing or phase shifting the expression of PER1 and PER2. These are called period 1 and period 2. So two of the important clock genes, and they reduce the expression of two of the other clock genes, ROR alpha and REVER B. So again, there's a direct effect on affecting clock gene signaling. At the same time, cortisol signaling through glucocorticoids can be inhibited by clock-mediated acetylation of glucocorticoid receptors, and also the binding of Cry1 and Cry2, this is called cryptochrome 1, cryptochrome 2, bind to the glucocorticoid receptor. So there's a back and forth. Why is that important? Because metabolism is regulated on a circadian rhythm. So you want your HPA axis, which is your regulator of metabolism, to be tied in with your clock gene. And this is also one of the ways that the brain can signal to the cells, because glucocorticoid is expressed on a circadian fashion, how this works. So there's a feedback back and forth telling each cell, hey, this is what's going on. We need more metabolism. We can rest for a while. We just woke up. All these kinds of things are happening all the time. Now, why would you want to use your stress response system, which could be thrown totally out of the loop when you get a call from the IRS? Why would you want that system to regulate your circadian rhythm? That seems a little bit, you'd be chaotic. So we have a large number of stressors that can generate extreme fluctuations in cortisol outside the normal diurnal rhythm. It can happen on any given day at any given time. Wouldn't this create havoc on the circadian rhythm? So remember I talked about this ultradian rhythm? You see these signals that come out from the brain. You have these little drip signals of ACTH. Those little differences, which are almost impossible to measure, you can't really measure them using, unless you're going to measure them every minute. So they're not really going to be a clinical, something that's going to be clinically beneficial necessarily. But that little ultradian rhythm is different when the brain is signaling a circadian signal versus a stress-induced signal. So even though it seems on the surface that these are the same sorts of signals, it's just ACTH, it really is a different sort of signal, and it allows both the cortisol production and the difference in the cells to know the difference between a circadian and or a stress-induced cortisol secretion. That's true if the stressor is acute. However, if the stressor is chronic, what ends up happening is you begin changing the adaptation to cortisol. Remember all 7 of those things that can affect cortisol signaling in the tissues? Those begin to adapt to, let's say, elevated cortisol levels, which then changes or desensitizes or creates a change in the cortisol signal, which then is affected or does affect the molecular clock and does affect the circadian rhythm. And so you slowly get out of sync. So it's a slower process. Thankfully, each time we have a stressful event, we're not completely thrown off our circadian rhythm, but over time, it does affect our circadian sensitivity, which then affects our metabolic efficiency, which then affects our chronic disease capacity or resilience. It's really important to note that there's 2 tissues that do not have glucocorticoid receptors, the suprachiasmatic nucleus itself and the pineal gland. Neither one of these are affected by cortisol, which is really good, because if they did, then we would really have no ability to reset the program. And so this is something that's really important to understand. So again, the feedback loop of both the suprachiasmatic nucleus and the pineal gland are cortisol independent, which is good for us. So here's something that can be very stressful. How about running a marathon? You can see on the right-hand side here, this is what happens to your cortisol levels when you run a marathon. You can see they shoot through the roof, and I can tell you that amount of cortisol will affect your immune system. It'll suppress your immune system. It will affect your circadian rhythm. It will affect your metabolism, and it will take a little while to recover. That's why it's very difficult to recover from a marathon if you're not ready for it. On the other hand, you can see very healthy people. This is on the left-hand side. This was looking at the diurnal rhythm of cortisol. You can see the cortisol awakening response at the beginning, that sharp peak. But depending on when these individuals exercise, whether it was in the morning or the afternoon, you can see a post-exercise cortisol spike. And in most people, you will also see a post-cortisol DHEA spike, which they didn't really measure here, but that's what you see. And so that's a healthy HPA axis. Exercise, physical activity is a stressor because it's a metabolic, it requires a metabolic response. So you should expect a metabolic response to a metabolic need like exercise. So again, these are all things that we need to be aware of that are influencing what we're measuring behind the scenes. And of course, this is a great review article talking about glucocorticoid circadian clock and how part of what's going on in Cushing's disease is this loop between excess glucocorticoid production and changing in their circadian rhythm itself. And the question is, which came first? And so there's this very early relationship between cortisol signaling, cortisol production, and clock gene in peripheral tissues, the influence going back and forth. And so this is sort of at the root of some of the chronic issues that we see with Cushing's or Cushing's-like conditions. So with this, I think it helps us start thinking differently about the HPA axis, thinking about it as an energy management system, not just a stress response system. Obviously, within that, we have this emergency override system, but that's really not the main function. That's the emergency, as it were, the override system. So personalized lifestyle medicine is really understanding, if you're going to understand all these signals, all the different lifestyle signals, whether it's diet, physical activity, stress, circadian rhythm, et cetera, and how those signals come in, you have to understand not only what types of stressors or what types of signals are affecting the body, but also the timing of those signals. And we don't have time to get into this, but as it turns out, timing of meals becomes very important for metabolic clocks, and you can actually get off of your signaling, your light dark cycle, by changing if you shift your meals by an hour, your peripheral clocks will start shifting, because one of the signals that they use is the amount of energy available, what food is available to them, and that will be affecting that as well. Lastly, I'll just mention briefly the other categories. I didn't want to just leave these aside, but glycemic dysregulation and inflammatory signals. Obviously, these are very important signals. TNF alpha, IL-1, beta, and IL-6 are some of the most potent HPA axis signals. So they will trigger the system to produce, obviously, for cortisol as an anti-inflammatory. And of course, this can come from anything. So obesity is an adipose-driven inflammatory condition. But if your patient has allergies or sinusitis or inflammatory bowel disease, or even like arthritis, all of these things are going to be driving the HPA axis to produce cortisol as a modulator for this, because inflammation can put a lot of stress on cells. The body wants to be able to benefit that. So let me just briefly talk about some of the testing phenomena. And I know, if you're listening to these in the archives of webinars that are on the Dutch library, are going to far exceed what I'm going to tell you here. But one of the things that I had talked about is, moving on from this. This is where everything was, 20 years ago, just taking the 4-point salivary test. It was a good start, but there was so many opportunities to expand. And the first was the fact that most of them didn't take advantage of the cortisol awakening response. And I know Dr. Jones has probably beat you over the head with CAR and understanding it. So she's done a much more comprehensive job than I'm going to do here. But remember, I talked about the idea that first morning cortisol is so important, because it tells you something that's going on with the suprachiasmatic nucleus. So oftentimes they were missing it. It was just being missed because the instructions were saying, take it between 6 and 8 a.m. or sometime after you wake up. And we just didn't have a good instructions on doing that. The reason I say it's so important is because, remember, I said the suprachiasmatic nucleus has a direct innervation with the adrenal gland. And basically what it's like doing is, as you've been sleeping, as it's dark, it's getting closer to morning, your brain is producing more ACTH, but the adrenal gland is actually not responding to it. It's being told, don't respond so much. So the cortisol is going up a little bit, but it's not going up a lot. It's slowly coming up as the ACTH is going up. And as soon as you wake up, you open your eyes, you see light, the suprachiasmatic nucleus tells the adrenal gland, okay, now is the time you should respond to this in a more robust way. And that's what we get, what we call the area under the curve, what we call the cortisol awakening response. It's the difference, the rising cortisol that occurs after you awaken. It's not just the total amount, it's the difference between awakening typically and 30 minutes later, something like that. We don't get this response at nighttime when you awake. You don't get this response when you take naps. And it really is a surrogate marker for the plasticity or the reactivity of the HPA axis. And of course, it's combining circadian rhythmicity as well as light sensitivity. And you want this to be robust. Obviously, if it's too robust, it probably means you're anticipating that today is going to be a stressful day, because it also has this sense of anticipatory stress. Now, I'm going to kind of go past this slide, but as you can see, there's huge review articles basically showing that this is the single most this biomarker is one of the most important single points used in stress-related research. And at one point, none of the labs were even offering the ability to do that. And so, I'm very thankful that Mark and his team have been able to incorporate this into their offerings, because it's really helpful. So people with elevated CAR are typically people that have higher life stress, higher job stress. Typically, it's higher on Monday through Friday than it is on Saturday and Sunday. So I remind people to take this during the week and not on the weekend. And it's lower, excuse me, it's lower in people with burnout, what we think of as true burnout, and post-traumatic stress. We typically see it being lower or flattened in many cases. So this normal curve, there's a lot going on in this normal curve that's there. So I always tell people, make sure that you know that it is a normal day, or that you're measuring the right thing. If somebody has a really bad day and they started taking their tests that morning, and something happened in the middle of the day that may not be typical. I mean, it could help you understand how they respond to that particular event, but it probably is not necessarily going to tell you, how they normally would respond. DHEA, I'm a big fan, although we don't know as much about how to interpret various DHEA. Should you measure DHEA, DHEA sulfate? As it turns out, I've written on this. They're really surrogate markers for one another. And so probably measuring one versus the other than maybe some subtleties in the research, are going to make probably a big difference. So, I think it's important. I'm actually very intrigued, because the research on DHEA, even though it was the darling of the industry in supplementing DHEA back in the 90s and the 2000s, we don't know as much about DHEA as we thought we did, primarily because in humans, DHEA is made by the zona reticularis, one of the layers of the adrenal gland. But in most of the research animals that we use to do our stress research, primarily rodents, they don't make DHEA in their adrenal glands, they make it in their gonads. And there's a different signaling that goes on there. So we really don't know why DHEA functions, how it functions. We don't make DHEA until we hit adrenarche, so around 8 or 9 years old is when we start making DHEA, and of course it peaks maybe in our 20s or 30, and then it begins going off. So there's a lot of things that we don't know. I believe that measuring DHEA especially cortisol to DHEA ratios can be helpful to really understand aging, tissue aging. So, I think the future of HPA axis testing, I think there's lots of things that are coming down the pike. Assessing HPA axis function and status, it's not always straightforward, as many of you know. I mean, you call in, you get help with some things. You really need to understand some of the nuanced understanding of how this as a surrogate marker reflects function. And I think we're going to learn more and more about connecting not only cortisol and DHEA, cortisol metabolites, timing differences, and actually also provocation tests. So provocation tests like dexamethasone or even ACTH to really find out what is the responsiveness of the HPA axis. And so I think stay tuned for more of that, because I think those require prescription, and that can't be done by everybody. Don't be afraid to question the test results if they don't match up with other signs and symptoms. You're always treating the patient, not the test result. So there's something, if things don't look right, or this is kind of weird, this doesn't look like they tested this right, or maybe they didn't, sample it right at the right time, or whatever, maybe they mix up their tubes, whatever. Really important to ask those questions, and maybe even ask what they were doing on the day they took this test. Did something interesting happen that they didn't, did they exercise at the wrong time, half an hour before they took their test? And also keep an eye open. I think, I'm really encouraged in talking with Mark and his team, and even some other labs as well, that are constantly asking questions. And I think they should, they're even challenging each other, several of the labs, and I think that's a really good thing in our industry. And it's going to be great for clinicians and their patients, that that there's a constant ongoing understanding of the literature and how we can interpret. Not everything in the literature can be immediately applied in the clinic, but we're able to add more and more things as we start thinking and being creative. So finally, I'm just going to introduce this idea because this is a huge area. People ask me, well, what are the strategies for supporting HPA axis? What do I do? If I'm not supporting the adrenal, what am I doing? Well, there's three areas that I think of: the supporting the central nervous system, supporting the adrenal, and I put that in there because there maybe are some things that we can do, and then modulating target tissue cortisol sensitivity. And you can see that I've divided this up. So what can we do to maintain appropriate hypothalamic responses? Well, modulate or reduce glycemic dysregulation, reduce perceived stressors, reduce inflammatory signaling, and increase circadian signals. So those, again, most of what we are going to do falls in this idea of these four key stressors. I can't stress enough, if I can use that as a pun, I can't stress enough the idea that if you can address these four key stressors, you are doing more for your patients than you are giving them anything, whether it's an adaptogen or even DHEA, or low levels of hydrocortisone or whatever. If you can get them to understand and get them to recognize and modulate how the brain is being inundated with stressors so that they can get about doing normal metabolism, you are going to have the most effect. Now, obviously, there are things that can be done: balancing neurotransmitters, perhaps neurosteroids. Consider supplementing some of the precursors, maybe DHEA. The idea of giving somebody pregnenolone, I've actually looked into this to find out, okay, what is happening. Likely, this is not going into the adrenal glands and then converting to cortisol and DHEA. Likely, how this is functioning is actually getting into the brain, acting as a neurosteroid. And by doing that, it does affect the HPA axis, but it doesn't do so in the way that we often think. And there's a lot of research, or I should say, there's a good amount of research on using oral pregnenolone for schizophrenia, primarily as a neurosteroid. And then there's other things that we can do. Adaptogenic herbs likely have an effect on the feedback, cortisol feedback mechanism, and they also probably have an effect on changing glucocorticoid receptor sensitivity, which is probably going to the target tissue side of things. So you can see here, if you can reduce inflammation, if you can reduce insulin resistance, reduce central adiposity, you can affect the cortisone shifting with the 11 beta HSD activity. So again, there's other things that you can do that will have an effect on cortisol sensitivity and cortisol effects that aren't, quote unquote, fixing the adrenals or fixing even directly the HPA axis. It's all of these other signals that I'm talking about. And certainly, perhaps, vitamin C, B vitamins, pantothenic acid, niacin, magnesium, zinc, these are all potential benefits for all cells. And of course, we know that a lot of these are concentrated in the adrenal glands. We certainly don't want the adrenal glands to become metabolically stressed under the need to produce lots of cortisol, or DHEA for that matter. So it's certainly something we want to support. It's an organ system that we definitely want to make sure is supported, but it's not the primary focus of fixing the HPA axis. So I know there might be a few questions out there. I think Amy maybe have gathered, but I appreciate you stopping in and listening to me talking about some of the things I'm very interested in.
Thanks, Dr. Williams. I've got a few questions here, and I'll probably just run through about 3 of them here.
Okay.
With your book, the second edition, is this available to purchase if you live in the UK, which I'm assuming it is if they go to thepointinstitute.org. Is that true?
Yeah, we will ship it to you if you want to pay the $30 that they take from you to do that, or you can just get the ebook. You can download it right away. So you can download, obviously no shipping for an ebook. And it's the ebook's a few dollars cheaper than the printed book. So yes, we send books to all over, and we send a lot to the UK regularly.
Okay, great. And that's just going to the pointinstitute.org, correct?
Yes, just go to the pointinstitute.org and go to our bookstore, and the other books are on there as well.
Great. And just for everybody that's still on here, we'll make sure we include that in our follow-up email tomorrow as well, Dr. Williams' website, which I know you all can see right here, but we'll make sure we reinforce that. Can you explain again, touch on how PTSD affects cortisol and the HPA axis?
Yeah, so normally when you have an adult, now there is a lot of differences between children with PTSD and adults. So I'm just talking about adults. We don't have as much clear understanding of what is going on with the HPA axis in children. And it really depends on their age and whether they're making DHEA, ironically. But in adults with PTSD, you will typically find low cortisol levels and more often than not, elevated DHEA. When I say elevated, higher than, let's say, the reference range. It may not be, elevated in a dangerous way. And what we think is going on is there's a hypersensitivity to cortisol in the brain, perhaps driven by neurosteroids or neurotransmitters or endocannabinoids, or just the fact that at a previous time in history there was a very high acute level of cortisol coming to the brain. And the theory is that this is a down regulation to protect all the tissues from excess cortisol. The brain tries to protect itself by reducing the signals that produce cortisol. And so these individuals become low in cortisol because of that. Now, you'd say, okay, well, that sounds good in some ways, but the problem is then they become less able to tolerate new stressors. So again, this is an adaptation with consequences. So that's at least one of the factors. The amount of information, I mean, there's surprisingly, it's frustrating because most of the time we get PTSD subjects, it's after the fact. So it's very difficult to have sort of progressive understanding of what's going on in the HPA axis before they underwent an acute stressor, and then what actually happens to bring them to the other side. And some of this is being done in the military. So we're seeing more testing being done in military so that we can find out what actually is going on in a stepwise fashion that changes the HPA axis. But unfortunately, we don't know as much as we'd like to.
Okay, and then this is going to be my final question here. Is there a connection, maybe you've seen, with DHEA and dopamine or any other neurotransmitters?
So yes, I mean, all of the neurotransmitters seem to be influenced by DHEA at some level because, like I said, the neurosteroid influence on GABA and glycine receptors probably more so than dopamine, but they're all influenced. So you see that effect on GABA and glycine is then going to affect the effects on dopamine. But I don't know that we have a clear understanding of how DHEA and dopamine receptors are related. I have not. That's probably an area I've not spent much time looking at, but I have not run across research in that area that often.
Actually, let me throw one more question because I think you might have some guidelines on this just based on, I know you've done so much research on nutrition as well with your background. I've got somebody asking a little bit, they're like, what's the best meal timing for insufficient cortisol production? Any thoughts on that?
Well, when you say, I don't know if, I would say it this way, it seems like the best meal timing, I mean, of course, meal timing is one of the most controversial issues at this point. I would say, I don't know that anybody's looked at meal timing for optimizing cortisol, because the question is, what is the optimal cortisol level? I mean, that's a whole nother question. But I would say my personal opinion, and based on the research that I've seen, there is a definite connection between the precipitous drop in cortisol after awakening and the body's need for, and I think metabolically, for eating. So eating breakfast, I guess is what I'm saying. Eating breakfast during daylight hours corresponds with the drop in cortisol. Well, remember, cortisol is a glucocorticoid. It's the reason that the cortisol is dropping is because it's assuming you're going to give it glucose. You're going to eat, and you're going to be your brain says, I'm awake. I'm now ready for the day. I need to start upregulating my immune system, so I'm going to drop my cortisol. I now need to eat to produce glucose. So I believe that eating during daylight hours is probably the key, and that goes to all the circadian rhythm information. And I would say eating early in the day. As far as the whole time restricted feeding idea, I believe that time restricted feeding should be done during daylight hours. So you should be eating during daylight hours, and probably, but I can't prove this, eating for more hours in the summer where daylight is longer and less in the winter is probably also normal. Um, but I don't know that anybody's tied that specifically with optimal cortisol levels. But I would say based on the biology, that would seem to be consistent.
Okay. All right, great. Okay, that's going to be it for today. Thanks, Dr. Williams, for being with us. Thanks, everybody who logged on. We had a big group with us today, so I appreciate everybody and all the questions. And just a reminder, we will send the on-demand recording along with the slides. I'll make sure to send the information from the Point Institute as well with regard to either downloading the ebook or ordering the book. Thanks again, everyone.